Hello everyone, this is Riya Bhatt and today we are going to talk about the another three organelles of the endomembrane system, that is Golgi apparatus, lysosomes and vacuoles. So as you can see, we will first go through Golgi apparatus. By reading the name itself, you would be wondering that what does it mean? But there not such a deep meaning. Instead, it has been named after an Italian scientist named Camillo Golgi, who discovered it in 1898 from the nerve cells of barn owl and some cats. It's located beneath the endoplasmic reticulum, and it can be defined as a network of membrane bound vesicles which are arranged parallel to each other. And these vesicles, which are stacked parallel to each other, are termed as cisternae. Each stack contains around 4 to 8 cisternae. It concentrically arranged around the nucleus for forming two phases, that is the cis phase and the trans phase, or the maturing phase. Basically, it works by packaging the materials which are formed in the endoplasmic reticulum and fuses with the cis phase of the Golgi apparatus and move towards the maturing phase. The vesicles in which it has been packed is termed as the transport vesicles, as you can see in the diagram. Proteins being synthesized in the ribosomes of the endoplasmic reticulum are being modified in the cisternae of the Golgi apparatus. In plant cells, they are found as single units or are unconnected units termed as dictyosomes. So, moving towards the functions of the Golgi apparatus, the main functions of Golgi are been modifying and sorting of the proteins, and so sometimes it is also compared to as the post office of the cell. This modifications includes phosphorylation and glycosylation of proteins and lipids both. Phosphorylation is adding up of phosphate molecules to the lipids and the protein molecules. Sometimes synthesis of protein glycans is also been carried out by the Golgi. Golgi separates the proteins and lipids received from the ER according to their destinations. For example, it also separates the digestive enzymes that are bound for lysosomes from hormones that will be secreted from the cells. It packages these materials into vesicles that are transported to the other parts of the cell or to the plasma membrane for the export. So, this was about Golgi body. Now, moving towards the other organelle, that is what is lysosomes. Lysosomes were first reported by a Belgian scientist Christian D2 who in 1955. Lysosomes are small membrane-bound vascular structures formed from the modifications and the packaging of the Golgi apparatus. The membrane of the lysosomes are surrounded by lipoproteins or are made up of lipoproteins, which is very much less permeable to the enzymes present in it. It contains some hydrolases in it, and the acidic conditions is maintained by pumping the protons into the interior of the lysosomes. The digestive enzymes which are being synthesized on ribosome, so that is of the ribosomes of the endoplasmic reticulum and packed into the lysosomes. And the portion of the smooth endoplasmic reticulum near the Golgi apparatus may also bud off like lysosomes. They are about 500 nanometers in diameter. Lysosomes are also known as suicidal bags as it contains some hydrolytic enzymes and are able to digest the cell. The hydrolytic enzymes which are present in the lysosomes are commonly nucleases, glycosidases, proteinases, and lipases. This hydrolase has an optimum pH of 5. These are found in eukaryotic cells except the mammalian RBCs and are absent in prokaryotes. Varied types of lysosomes are found, such as primary lysosomes, secondary lysosomes, tertiary lysosomes, and autophagosomes. There are many functions of the lysosomes. Amongst them, the main one are extracellular digestion. Extracellular digestion is a process which is reversible to the phagocytosis, and enzymes are liberated outside the cell in the extracellular digestion. It occurs during the penetration of sperm in the ovum. Cellular digestion. It occurs either in pathological conditions or when the cell dies, and it is known as autolysis. Digestion of the foreign materials, that is engulfing or phagocytosis of the foreign bodies such as bacteria. Waste disposals, that is, it is also a cellular. Moving towards our third organelle of the endomembrane system, that is what is vacuoles or cytoplasmic vacuoles. These are the non-cytoplasmic areas present inside the cytoplasm, or rather fluid-filled membrane-bound organelles, which are surrounded by the double membrane, which is known as tonoplast. And this double membrane is a semi-permeable membrane. They occupy around 5 to 90% of the space according to the cell type. And it occupies a major volume of the cell in the plant cells. They are being classified into 4 types depending upon the contents and the functions they perform. Among them, the first one is the food vacuoles. These food vacuoles are found in the protist for engulfing the food. Second one is contractile vacuoles. These are found in the protist like amoeba, which takes part in osmoregulation and excretion. Third one is the sep vacuoles, which are found in the plant cells. They help to store and concentrate some mineral salts as well as nutrients. And the last one is the gas vacuole or the air vacuole, which is found in all prokaryotes. And they are not only stored metabolic gases, but they also helps in maintaining the buoyancy of the cells. So accordingly, it functions for water storage. It maintains the turbo pressure and hydrostatic pressure, performs endocytosis as well as exocytosis. It maintains the acidic internal pH, contains waste materials and excess storage materials. Thank you so much. If you are having any doubt or any suggestions, please comment in the comment section. Thank you so much.